Now the next organelle that we are going to take into account is cilia and flagella. These are hair-like projections which are present on the free surface of the cell, as you can see over here. These are whip-like organelles basically. You understand what is a whip? Whip basically, whip is the thing you must have seen in movies or somewhere else, which is used in the form of a weapon which is used to assault people. You must have seen the villain basically uses it like this and then hits people with that. Just like you put a thread on this, supposedly this is the handle of something and you put a thread and you move it like this, the movement that would be created would be whip-like. So I hope I am able to make it clear to you what is whip-like. So these two organelles are whip-like structures which are present on the free surface of the cell, and what is their function? That is bringing the motility to the cell, that the cell is able to move within its surroundings. That is brought about by these organelles. Alright, now please remember, if the cell is unicellular, then the movement brought about by these organelles would be the actual movement of the entire organism. But if we are talking about multicellular organism, there is movement which is to be brought about, like in the case of macrophages. There are cells which have to go and patrol within the entire blood of human body, and they have to look for the bacterial cells and then engulf them. So that motility sort of thing would be brought about by these organelles. So we take into consideration flagellum and celium. Flagellum is singular, celium is singular, and Flagella and Cilia are their plural forms. Now as you can see over here, this is a 3D representation of an electron micrograph for a cell, where you see long whip-like structures. Again over here it is a whip-like structure, slender structure, very long. This is flagella, and the next that you see is the simplest diagram which you were introduced to in your class 6th or 7th, I guess, the diagram of paramecium that we make. This paramecium is having small cilia. If try to recall how you make the diagram of paramecium, you put small hair-like projections over there, very small hair-like projections in the periphery of the diagram, and that you can see over here. So these are flagella and cilia. First one that I showed you was flagellum, these long ones, and the second one, both of them are at the same level of magnification, and the smaller ones are celium. Now what we have to see when we understand these both is, first is the point of difference between the both. What is the difference between both and what is the similarity? One thing you have to remember, they might have difference in their movement, in their level of the differentiation that they show, but their ultrastructure happens to be similar. Alright, you have to remember that the size may vary, the basic movement that is brought about by the organelle, both cilia and flagella, it could vary, but the ultrastructure is similar. So they have differences as well as similarities. So first of all we are going to see the differences. Now what is the first point of difference is that cilia and flagella, their length is different. Now what is the length? What is the length difference that we have to deal with? When I talk about cilia, it would be from 10 to 50 micrometer, not more than that. Okay, this would be the length of cilia, quite small it is. And if it is about flagellum, it would be around 100 micrometer to 200 micrometers. So you can imagine the length difference. As you can see over here, this celium is not longer than, you see, 10 to 50 micrometer, and comparatively see the flagellum thing, it is quite longer, that is 100 to 200 micrometers of the length can be attained in a flagellum. Now coming next, what we have is that their movement is different. Alright, for understanding the movement part, you need to consider flagella differently and the cilia movement differently. Now first of all, we see the movement which is brought about by flagella. Now see, there is a whip-like structure, we know that. When that whip moves, basically, if that whip is being moved like this, what is being created? There are ripples which are being created. Have you ever thrown a stone in water or something falls in water? You must have observed there are ripples which are being created. You study ripple effect in physics as well. So that creation of ripples is brought about by these organelles, and there is an undulating fashioned movement which is brought about by creating the ripples. When the ripples are being created, the cell is able to move further. Alright, this is how the movement is brought about. That ripple thing moves the medium and the cell moves further. Now there are two types of movements, the helical and the planar waves, which are created because of the ripples being generated. They are able to move the cell in the forward direction or in the backward direction. Now you have to see that the flagella being a long whip, it is having the waves which are created in such a fashion that it could move from the base of the flagellum to the tip. Supposedly, each one of you must have seen some sort of 3D representation or some sort of animated representation of a human sperm ever in your life. You must have seen that human sperm is like this and it is having a long flagella along with it. Now this long flagellum, there are two types of movements which could be brought about. First is that this flagellum is moving and the ripples are being created in such a way that the waves are being created from base towards the tip. Alright, the waves are being created from base towards the tip. Supposedly this is my finger and I am starting the movement from the base towards the tip. Alright, I could have represented it in a better way using a thread. You can also use a thread to understand it, that if you start the movement from the base towards the tip, what happens is helical waves are created in such a manner that the cell is pushed forward, that the cell would move forward. Alright, and in the other case, when it is from tip to the base, supposedly the movement starts from here, over here, like this. It starts moving, this part starts moving and it goes towards the base, then the cell would be pulled in the medium, in the surrounding medium, that would be liquid of course. So this is how the flagella is able to generate its movement. Now the second thing that you need to learn is that how many types of flagella are there. There are two types of flagella. First one is tinsel type of flagella and the second one is whiplash type. Okay, so we have tinsel flagella and we have whiplash flagella. What is there in tinsel flagella? That there are flimmer filaments, small filaments which are present. I would write over here because this is a term you need to remember, because this is the basic distinction, this is the basic character that brings about this classification, that flagella can be classified into tinsel type and whiplash type. The flimmer filaments, they are responsible for a type of movement which is opposite to this, that the cell, when it has to be pushed, it would be pulled. Now try to understand it. Supposedly, now that names are clear, I would represent it over here. And see from here as well. This flagellum, longer one, it would have short finger-like projections again, yet again, that would be flimmer filaments. And when the flimmer filaments are present in tinsel type of flagellum, what would happen is, like, consider this as flagellum, now a thicker one as in we are seeing it to a higher level of magnification. It would have small flimmer filaments over it. Now what these flimmer filaments are able to do, that when the movement is from base towards the tip, we expect the cell gets pushed, but over here the cell would get pulled in case of tinsel filament, and the other one would be the naked filament, that is not having any flimmer filaments, and it would be whiplash. So this happens to be whiplash and the other filament happens to be tinsel type. Alright. Now this was all about flagella, its classification, it's on the basis of the filaments which are present. Next what we see is how does cilia show the movement. Next is that the cell has to undertake all the movement in the medium with the help of these small projections, very small they are as compared to the longer one, that is flagella. Now you must have seen that, ever seen somebody using an oar and moving in a boat. You might have seen somewhere in the print media or popular media that boat is being propelled using oars, and if the boat is quite long, there are many oars which are being used. Many people sit with oars in their hands and they propel the boat further. So it is the force which is being generated by the oars which propels the boat further. So this is what happens in the case of cilia as well. Supposedly this is the free surface and there are these cilia which are present. So cilia accomplishes its movement in two strokes. First one being power stroke, okay. The propulsion of the cell forward or backward is brought about by the oar-like functioning of the cilia. The first stroke being power stroke. Supposedly the cilia are being moved in this direction, they are moving. The direction of movement would be in this direction. Please remember this. Okay, the power stroke is in opposite direction. That means the cilia themselves, they move in the opposite direction, and direction of the movement happens to be opposite. Now in order to bring, now again remember how the oars are moving, how the oars are used for propelling. First the person pushes them for backwards, and then it brings back the oars, and then again it brings the movement by pushing them backward, basically. I'm sorry. So what is next to be done is the second stroke would be recovery stroke. I'm going to repeat it because I just guess because of my fumbling you might not have understood it. You must have seen people, they are rowing the boat, they are using the oars, they push the oars, then they bring back the oars in the same position, then they push that again, then they bring that back again. So basically they are recovering from that pushing whatever has been created. Same is in the case of the cilium, because it is similar to oar-like function. Again, this is the free surface. The cilium, these are cilia basically which I have shown you. The cilia which had moved in this direction, they would come back to they would try to come back to their original position so that they are ready for another power stroke. So this particular movement of cilia in the beginning, they get folded, they are trying to come back to the condition where they can go back to the power stroke. So this particular stroke is known as recovery stroke, and as you know, when the boat is moving, basically when the recovery is being done, the movement is that of the previous propulsion. So the movement is being generated by power stroke, and the recovery stroke is in order to bring back the cilia into their original condition so that they can take another power stroke. Now the next thing that we are going to study is in terms of flagella, how many types of flagellum are present in cells, and what is the classification of cells on basis of the presence of flagella. So the first one is, as you can see over here, the diagrams are quite descriptive. There is the type of flagella which is known as Peritrichus. Peritrichus, as you can see, the flagella is uniformly present all over the free surface of the cells. This is the cell body, and the free surfaces all over the cell is having flagellum. Atrichus represents the cell which is not having any sort of flagella. Monotrichus would have one flagella. Amphitrichus, amphi means both, alright, as we use in the word amphibian. They are able to survive in both the land and terrestrial and aquatic conditions. Ambidextrous, that means somebody who can use both the hands. So amphitrichus is at both ends you are going to find the flagella, and in the case of Lophotrichus, at one end there could be two or more flagella. So instead of one, which was in the case of monotrichus at one end, Lophotrichus is the condition where we have many flagellum which are located at one end. So this is all we have to understand about flagella and cilia. Next we are going to see the ultrastructure of cilia and flagella, which I told you in the beginning that it happens to be similar in both the cases, despite of all the differences which they have in their functioning and in their length. Their ultrastructure happens to be similar, and that is what we are going to consider next.